Hi guys, Miss Francis here. Last unit was all about DNA as the molecule of life. In this unit, we're going to look at how life is changing because of DNA. So genetic engineering, what is it? It is exactly what it says. It's the engineering of genes, where the direct manipulation of genes for practical purposes is what scientists do when they're genetically engineering materials. Another way of saying it is that the manipulation of organisms or their components to make useful products. Now, why would scientists care about genetic engineering? Because that technology, that DNA technology, is now applied in agriculture, in criminal law, in basic research. So it's applicable in a wide variety of industries. So for example, in agriculture, maybe I could grow a plant that's resistant to a pest. So now I don't have to spray pesticide. That plant already has that resistance to the pest. Criminal law, for example, and forensics. But its most important achievements are in basic research. So in order to study a particular gene, I would do something called gene cloning. And gene cloning is exactly what it is. Cloning of a gene, so making multiple identical copies of gene-sized pieces of DNA. Now, why would I want to focus on just one gene? Well, let's say I want to look at sickle cell anemia. I only want the gene that codes for sickle cell anemia. Once I isolate that gene, then I want to be able to clone it so that I could do research on it over and over and over again. Now, most methods for cloning pieces of DNA share certain general features. For example, some sort of foreign gene, that's the gene of interest. In our example, it is sickle cell anemia, the gene that codes for that disease, is inserted into a bacterial plasmid. And a bacterial plasmid, the plasmid, remember, is that small circular DNA that can be found in prokaryotic organisms, and a bacteria is a prokaryote. Now, once this foreign gene, this gene in our example that's coding for sickle cell anemia, is inserted into that plasmid, it's now called recombinant DNA because it's a recombination. It's got the foreign gene and now it's got the plasmid. So what would this technique look like? Let me show you by annotating this image. So here we've got our bacteria, and our bacteria has a plasmid, where that plasmid is a small small circular piece of DNA. And now we've got our cell containing the gene of interest. So here's the DNA found in the nucleus of that cell. This is our DNA, and this little black part that I just covered up is our gene of interest. This is the gene that codes for, following along with our example, sickle cell anemia. So first I'm going to isolate the plasmid and isolate this gene. Then I insert that gene into the plasmid, and now I have created recombinant DNA. Then that plasmid is put into a bacteria cell. Now I have created recombinant bacterium. And then this bacterium that contains our plasmid, that contains the gene of interest, so our recombinant bacterium, is going to be cloned. Then scientists are able to identify the desired clone, where ultimately they can look at the copies of the gene that were made or the copies of the protein. So let's say scientists are interested in looking at the gene. They might look at the gene that codes for pest resistance, like I mentioned earlier. And now plants will be grown with pest resistance, so I don't have to spray a pesticide giving them that pest resistance, which could result in environmental issues. That plant already has that pest resistance, so now that pest won't eat the plant. Or maybe I want to look at the proteins that were created. For example, I might look at human growth hormones that may then be used to treat individuals with stunted growth. Or maybe I want to look at copies of proteins, the protein insulin, for example, and be able to create artificial copies of insulin to give to people who suffer from diabetes. So there are two potential uses of cloned genes, and they're going to fall into two general categories. So the first goal, or the first category, is to produce a protein product. For example, like we mentioned, a bacteria that carries the gene that codes for human growth hormone, because remember, human growth hormone is a protein, or insulin. Insulin is a protein where I can then produce large quantities of the hormone, of human growth hormone, to treat stunted growth, or large quantities of insulin to give to people who suffer from diabetes. Alternatively, maybe I want to look at the copies of the gene itself. Now, this is where I was talking about the gene that could be inserted into a plant for pest resistance. But more importantly, it enables scientists to determine that gene's nucleotide sequence. So taking it back to our example that we started with, I could find the nucleotide sequence for sickle cell anemia. And now, using DNA technology like CRISPR, maybe I could go back and fix the mistake that was made in the nucleotide sequence that's giving that person sickle cell anemia. Another application would be maybe I could look at the gene that causes a certain form of breast cancer, and fix the mistake in that gene that caused that person to have breast cancer. So next time, we're going to look at restriction analysis, which is one of the basic tools that is used in this DNA technology.